at the Genetics and Genomics program. She earned her MD in 2023 from the Monterey Institute of Technology in New Mexico. Her research background includes tissue engineering at Harvard Medical School and cancer immunology at the National Institute of Medical Sciences in Mexico City. She joined the Lechinier lab last year, where she now focuses on cancer genomics and uncovering mechanisms of treatment resistance. Please join me in welcoming Margot as she presents her work on clonal selection in treatment of minimal residual diseases in chronic lymphocytic leukemia.
Hi everyone, and thank you for a nice introduction. As Ali said, I'm going to present my project. It's mostly the framework of this project titled Clonal Selection in Treatment of Minimal Residual Disease in Chronic Lymphocytic Leukemia. And I'm just going to start.
You want to use the microphone?
Oh, yeah.
Just help her.
Yeah, you just.
Sorry about that.
Yeah, turn it on.
Cool. So, yeah, I just said that I'm going to present the framework for this work. And I'm just going to start off by stating that all of our cells in our bodies develop somatic mutations. And I want to highlight two different types of mutations. We have germline mutations here. And these germline mutations come from our parents. So either in one or both of the parental gametes, they already have that mutation happening, and then that mutation passes on to the embryo and the living organism, and that mutation is expressed in every cell of their bodies because it comes from the germline, meaning their parents. But we also have somatic mutations, and these are the ones that I'm going to be focusing on from now on. And the somatic mutations don't come from the parental gametes, but at some point during the embryogenesis, or even after birth and during our lifetime, we can develop somatic mutations. And these somatic mutations aren't expressed in all of the cells in our body, but only in one particular affected area. Meaning, for example, if you're exposed to UV light or sun, your skin can acquire all of these somatic mutations, and those mutations will only be expressed in that affected area. And we care about that because cancer is a disease originated due to the development, accumulation, and selection of somatic mutations. And here, I just want to highlight this timeline of how it works. Here we can see the embryo. Most of the cells are lilac and some of them are pink. The pink ones represent embryonic mutations, meaning that happened during the development. But as the individual is born and then grows, you can see here that during early life, like pretty much it stays the same. So the lilac cells are the higher proportion, and then the pink ones are still there. Like you can still see both the unmutated and the mutated cells. And then, for example, here you can see this individual as a young adult, and you can see the purple cells, which are transient. Like it acquires that mutation, but then that mutation might not be even fit to further progress in the individual, so those cells regress and you get back to this state of like lilac cells. But then as you further age, you still acquire, like with different environmental exposures and also just inherent to life, you just acquire more and more mutations throughout life. For example, here you start seeing a bunch of different, like it's not only lilac anymore, but you can see a lot of different colors representing different clones and different mutations in these cells. And even here in the dark blue one, you can see the emergence of a cancer clone. So in this work, I'm going to be mostly talking about a type of cancer that is chronic lymphocytic leukemia, or CLL. This cancer is the most common type of leukemia in adults. It accounts for 1% of new cancer cases in the U.S. It affects primarily males, and then the average age at time of diagnosis is 70 years. And even though there are a bunch of therapies and the therapies do work in these patients, there's currently no cure available, and once those treatments are used, like there are no more options that you can give to those patients. So we're very interested in further understanding the mechanisms of treatment resistance in this cancer. Now, zooming in a little bit more into CLL. CLL originates from an immune cell that's called the B cell. This B cell has different functions throughout our life. Like a normal B cell has the capacity of producing antibodies, of presenting antigen to other immune cells, and also this cell is very important in immune memory. For when you get a cold or flu, like these cells are the ones that remember all of that information and then will attack as soon as they see a pathogen entering your body again. And these cells do their function through something called a BCR receptor. A BCR receptor is an antibody-like structure, like we can see here in the surface of this little cell. So these antibodies recognize antigens or pathogens that can be harmful for us. And then through this BCR receptor, they can be activated and then produce more antibodies or activate the immune system through further mechanisms. So I would like to start explaining the pathogenesis of chronic lymphocytic leukemia through the start with an A B cell. So we have here B cell that hasn't been activated or anything, hasn't encountered antigen, and it can be activated through two different pathways. The first one is, so here you have the B cells, they encounter the antigen, and then they're activated. They're activated with the help of other cells called T cells. These T cells further help the B cells to mature and mutate their BCR to start producing antibodies that are much more high affinity and to kill the pathogen in a much more efficient way. So with this T cell activation, you give rise to an antigen experienced memory B cell. These B cells are fully differentiated, they're long-lived, and they're the ones that keep the memory in our bodies from these pathogens. We also have the T cell independent immune response. So these B cells can still encounter antigen and react to it, but then without the T cells that are helping them here, this response will be much more less in affinity. So this will still produce antibodies, but the antibodies will have lower affinity, and these cells won't live as long as the other ones that were activated with the T cells. In both the scenarios still, like if you acquire environmental damage or mutations in the BCR, you can still experience something that's called monoclonal B-cell lymphocytosis, and this basically just means like from the normal number of B cells that you have in your body, you have an increase in that number, and that is not necessarily pathologic, but then some of those patients will progress if they still have some other damages in their cells. They'll still progress into CLL. So we have the two types of CLL that I'll be talking about today. The first one is the mutated CLL, so meaning the BCR receptor was mutated through the help of the T cell, and from now on I'm just going to refer to it as mutated CLL, but know that the mutation refers to the heavy chain of the BCR receptor. And then the unmutated CLL, meaning that the cell was activated, but then it wasn't further mutated they didn't have the help of the T cells. So just want to highlight here that the origin of B cell can be very different, and depending on the cells, it can be originated in different stages of B cell maturation. And we care about this because when we look at it through a mutational profile, here on the y-axis you can see the unmutated CLL, and on the x-axis you can see the mutated CLL. And you can see that the mutations don't really, like a lot of mutations don't really overlap. There are some others that do, but this just tells you that these two cancers behave super differently. And this work, they also reconstruct the time in which these mutations happen. And so here in purple, you can see the mutated CLL and the unmutated CLL. The first thing that I want you to notice is that the unmutated CLL acquires much more mutations. And the other thing that I want you to notice here is the timing in which these mutations happen. So for example, here you can see the trisomy 12 is the first one that happens in both of them, but as you see down below, like for example, I want to highlight here BRAF. The BRAF mutation occurs very early in the development of mutated CLL, but then it occurs very late in the unmutated CLL. So this just tells you how like the disease progression of these two different types of CLL is very different. And when we look at it through the lens of clinical outcomes, we can see that the unmutated CLL is much more aggressive than the mutated CLL, meaning like the patients with mutated CLL have more favorable prognosis. I've been talking about therapies, and so I just want to take a quick note. So the treatment of CLL had been from the 50s till the 90s. It had been chemotherapy, but then in 1997, the first monoclonal antibody was developed, and this monoclonal antibody was rituximab. And from this, we started the era of targeted therapy, and this included as well also CLL. So you have BTK inhibitors, BCL inhibitors target the apoptotic pathway of the cells, and you can see here that in comparison with chemotherapy, this Kaplan-Meier curve, the patients with that received targeted therapy did much better than the ones that only receiving chemotherapy. And this, then I want to highlight how this progresses in a patient. So here, as I've mentioned, you have your normal circulating B cells, and they're in charge of your immune response. Some mutations will end up in the patient acquiring monoclonal B cell lymphocytosis, and then it'll progress to CLL. Then the patient might even be asymptomatic. And here at this point, treatment is not even necessary. So because the therapies are so limited in these patients, like clinicians will most of the times just delay treatment as much as possible till the patients start developing symptoms, because otherwise the treatment will be very difficult and the cells will mutate a lot, that it will be uncontrollable, and the patients will end up doing worse than if they're treated before in their clinical progression. And so when they start developing symptoms, like for example, it can be unintended weight loss or node swelling, night sweats, then it's then when they will start therapy. And if we see it from a molecular point of view, we can see the founding clone here in yellow. So all of the clones of this CLL will have this yellow mutation in a random gene. From then, and then as time progresses, further subclones can be originated. For example, here the green one, it acquired another mutation and in another gene, and for example, the blue one here even got a deletion of a whole arm of a chromosome. So as you progress, you start, like, the disease starts getting much more complex. You not only have one mutation, but then you have like this heterogeneous population of cells that can behave very differently when you treat them. So, for example, here after therapy, I've mentioned therapy can be very effective. So you have all of the collapse of this cell populations, but then at some point it can be months, it can be years. There's no way of predicting this, but the cancer or CLL will most likely return. So here we have the relapse CLL, and then you have different cycles of therapy, and then it will end up relapsing and then complicating more the disease. And it just mimics the same in the molecular landscape. Like, for example, we can see here that the therapy was very effective in both against the yellow and the green clones, but then the blue clone somehow survived for like those months and years, and then it'll end up growing again exponentially, and that blue clone will give rise to other subclones and making it way more difficult to treat. A paper by my PI and other colleagues characterize the growth dynamics of this cancer of CLL. So they have serial samples of different patients and then characterize the growth of each one of these leukemias. So for example, you have 3 different types of growth: the exponential growth that we can see here in red, the logistic growth that also grows but not as much, and then it just kind of plateaus after some time, and then indeterminate growth, which is kind of in the middle of both. And when we see it from a mutation standpoint, you can see here that the populations are very different. For example, the logistic ones, when you see the mutated or unmutated status of the CLL, you can see that the majority of patients with logistic growth had mutated the mutated receptor. And when you see the mutations that they acquire, they have they do have mutations, but very low number of mutations here. And when you see the exponential, you see that most of the cases are accounted with the unmutated type of CLL, and that they acquire much more mutations per patient, meaning like this cancer, like this accumulation of mutations, gives rise to this much more aggressive cancer in these patients. And so you can not only determine the growth in the whole CLL itself, you can also determine the growth through each individual clone. So here, for example, you have the founding clone of the CLL, which gave rise to another subclone in black. So here you can see through time how this black clone didn't really grow. It stayed steady state. Then if you have other more complex cases, you can also see, for example, this common ancestor, the one that originated the CLL, it divided into 2 different subclones, and then the subclones have other subclones. But I'm gonna focus on this on the right. So you can see here that the orange and the purple clone and even the dark clone have an exponential characteristic, an exponential growth, whereas the other ones are just declining. So you can very well characterize how these clones are behaving, and for this, that hasn't been done in something that's called minimal residual disease that I'll touch upon further. But so minimal residual disease is this point in between therapies in which the CLLs are present in the body, but they're not detectable by traditional blood tests, or otherwise the patient will be without symptoms, basically in remission. It's in that period while they acquire treatment resistance. And so we want to target this minimal residual disease state. So there are several tests that can be done currently that the patients can have and know, oh, how is my cancer progressing? So this current test can be either be done by flow cytometry, by PCR, or sequencing of the PCR receptor. And this basically just tells you whether or not you have still cells present in the body. Most of them have false positives, and the error rate is not even that great, so they can detect 1 cancer cell in 10,000 cancer cells, and the result is either positive or negative. They don't say anything else about the mutations of the current status or how the cell might behave. So what we want to do is target this minimal residual disease state of the patients and do something that's called duplex DNA sequencing. Duplex DNA sequencing is a very sensitive way to do DNA sequencing. So the current way of doing DNA sequencing, you just acquire the information of 1 strand of DNA. With duplex DNA sequencing, you acquire the information of both strands of DNA. So I'm just going to go over it, through it a little bit quickly. So we have the top and bottom strand that have been barcoded, prepared, and adapted and everything, and on each side you have the blue and the orange barcode, so that's how you will find it. And then you go through a regular sequencing run, and then after with the computational processing, you see all of the repeats and amplification reads that you get. So here we can see the top strand, and you can see, for example, here the green, all of the amplificated products from the top strand have the green, but then the red and the purple dot are most likely sequencing errors. because not all of the amplification products have it. But then when you see the bottom strand here, you can see that all of them have both of them, like both the green and the brown. So what we can do here, we can do something that's called single strand consensus. So we see that all of this strands, all of the top strand has the green mutation, and then all of the bottom strand has both the green and the brown. What duplex sequencing enables us to do is to identify true mutations by doing something one step further. So we can do double strand consensus so that we can identify the one true mutation that would be the green and like discard all of the other ones that wouldn't be caught otherwise. So this method allows us to get an error rate of 10 to the minus 6. So in other words, getting one cancer cell per 1,000 cells, or in sequencing terms, get one true mutation every 1 million bases. So our goal with this project is to understand the minimal residual disease clones in chronic lymphocytic leukemia using DNA sequencing to better predict treatment resistance mechanisms. Yes?
Can I ask a question about that methodology?
Of course.
What kind of mutations can this procedure detect? Is it just single nucleotide transversions, transitions, or how does it handle insertions and deletions? And is it also limited to the size of the insertions, deletions, as mutations?
I think it would also be related to the length of the read, but you can also detect like copy number variations and like not only single SMPs. Yes, I can further look into it and give you a better answer.
Okay.
Yeah. So with this method, we want to monitor the emergence of the resistant clones during the minimal residual disease before they start growing again exponentially. We want to characterize the growth kinetics and fitness of each resistant mutations during sequential targeted therapy at a very deep level and a very low level of disease, so meaning like 1 cell every million cells, and test the therapy effectiveness or monitor the therapy effectiveness. And then we can also do the phylogenetic reconstruction of like, which subclones arise from what clone on the diagram that we saw before. So some preliminary results that my lab has done before. This is the measurement and follow-up of the CLL populations of one patient. So each on the y-axis here we have each time point, and then each color is a different clone in the patient. Here we're seeing a total, like out of the sample that we got, the abundance level was 1.01% of total cells, meaning that we could detect the cells in the MRD state. And we got a very deep coverage, around 100,000x raw coverage, which translated into a 10,000x duplex coverage. And we can see very clearly the growth kinetics of each clone. When we look at it through another lens, we have data from another patient that was receiving BTK inhibitors and different therapies throughout their treatment regimen. So each different line here is a different clone. So for example, I just want to highlight the green clone here, how even with the treatment, it just kept growing, and then after these CAR T cell therapy, like it went down. And so you can very easily and very effectively track the growth of these clones. But the other clones that I want to highlight here are all of the red ones. So you can see here after this CAR T therapy, here the patient had the rise of these red clones. These red clones all of them have different mutations, but those mutations are all in the same codon, and then they all give rise to the same mutation. So they all change a cysteine for a serine. So you can even if the mutation was different, like they all have the same product, the same amino acid. And when you see the growth fitness of this clone, you can see that the three of them behave very similarly. So yeah, what we want to do with this duplex sequencing in MRD is to be able to track all of these clones in a very low level of disease. And with this, I just want to conclude this talk making a summary that CLL is a biologically complex disease with variable outcomes depending on the subtype. Despite the advances in CLL, CLL frequently develops treatment resistance, and the duplex sequencing enables us to monitor sensitively the MRD subclones, aiding in the detection and treatment resistance and disease progression. And lastly, I want to highlight some future directions in which we want to take this project into. The first one is we want to characterize the mutational profiles associated with different treatment regimens, creation of a pipeline to predict the potential subclone behavior before it even starts growing. So what type of growth it will be, like either logistic, exponential, or indeterminate. And then monitor the disease using not only genomic CLL DNA, but also cell-free DNA, which can give us much more information and insights to what's happening to other cells in the body. And then characterize these subclones through methylation profiling too. And with that, I just want to thank my lab, thank the sequencing core at BU and the program of genetics and genomics, as well as Jennifer Brown's lab at Harvard, from which we get the samples, and the GSI for hosting this stuff. And I would like to open the floor for any questions. Thank
you. So there was a specific amino acid mutation coming on the rise, so to say, right?
Right.
In this population. So, so this information would allow to prepare for it, or how would you basically, what the value of that? So there will be some specific treatment that can.
Yeah. So we want. Yeah. So through the characterization and having all of the serial samples, like even when the treatment is working and we have this MRD state, we want to monitor that and at some point like create this pipeline in which we can predict like, oh, this patient like with this therapy might end up in exponential growth, logistic growth, or might just end up like in this remission state for years. So it just gives like the clinician more tools to see like, oh, do we need to start therapy before, or even like to see. I know that they're also working in targeting this MRD with antibody development, or like targeting this specific cells and trying to kill them before they start growing again. So that's like the overall long-term goal of this. But yeah,
so if that amino acid is on the surface, right? So
yeah, exactly. Like probably we can target the surface protein hitting. Yeah. Yes.
I might have missed this, but you're sequencing the DNA from these CLL and MLL patient samples,
Yes.
and then comparing them back to what genome as a reference, or using the patients?
We have both the normal genome and like the normal patient's genome and the CLL.
What are you using as the normal patient's genome? Let's say on
I'm not sure.
What are you using that to define your germline background versus your somatic mutations?
Right. I'm not sure in this case, honestly, but I know that regularly they use blood, but since it's a blood cancer, it cannot. That cannot be. But I can. Yeah, I don't know which.
Saliva?
So this is whole genome sequencing, is that right?
Yeah, but yeah, right.
And so the duplex DNA is a way to eliminate anything that's a PCR error or sequencing error from your Illumina, essentially.
Right.
Yeah, that's right.
Yeah.
So do you ever see changes in the immunoglobulin genes? Do those change over? Does the IgH acquire mutations as well, or is that, or do you mostly seeing things elsewhere?
What I know about the IgV, like, I'm not sure if they mutate over time, but you can know which cells are the malignant ones through the mutations of those the IgVs there. But I don't quite know if they further mutate like
Can you resolve those rearranged IgH regions from your data?
rearrange meaning
So when you have the mutated IgH, then determining the sequence of those is difficult because a regular aligner doesn't find a single unique site for that because the IG is highly variable and they're very deployable. So are you able to identify the clonal IgH in your
we didn't
data?
do it for this project, but I know they have another project in which they only sequence the BCR, so they put down the BCR and they do that, but not for this one. Yeah. Yes.
Have you also thought about in your sequencing to look for translocations? That's a common event in cancers, you know.
Right. Yeah. Well, we can also look at that, like the copy number variations and translocations, but like I don't think we've looked at it with this data set yet.
The duplex DNA method won't capture translocations unless you change your
long read, yeah. I don't think we've done it for this project, but we could because our lab we have also a nanopore, so we can also do this duplex sequencing with the nanopore and probably account for that.
Just one more thing more on the cancer biology. Looking at that, the time course of the different clones that you see, can you actually identify clone-specific responses to the different agents, even generate enough data to be able to analyze that?
That's what we want to do. Yes. Yeah. Kind of difficult. Yeah. But yeah, that's what. Yeah.
It's kind of just thinking of your figure. It's sort of surprising that you basically have one clone that stays fairly high and doesn't seem to really
be
affected
affected by anything. Yeah. Right. Yeah. Yeah. With any treatment, it's still there.
And I mean, is that definitely a CLL clone rather than?
Yeah. Yeah. All of the clones depicted here are CLL.
Is there a way to tell after certain therapies that we predict that these are the clones that are going to be more resistant, or is it random?
Not right now, but that's what we want. Like, yeah, measure the mutations after each therapy and then build a pipeline then. right now, I think our patients, the patient samples that we have are around 20. But yeah, we might take more. Well,
This is this the I can't remember quite enough about BTK, but is that the active site serine that the group is reacting with? This cysteine 481.
yeah,
So that's kind of a known and validated resistance mechanism that you see.
Do you think this could inform the order of treatment that a patient would receive so that it would minimize the number, or you could like pull it down to a single or one type of clone that you know how to treat?
I think at some point it just gets so complicated that that would be one of the end goals of this. But yeah, we hope to give the clinicians further information so we can have like this consensus of like probably even chemotherapy is best at the beginning and then just like treat with this targeted therapy. Yeah, I think that would take time.